The Enterobacteriaceae are a group of medically important gram-negative rods, and in box 25.1 we have a number of different species listed. These bacteria are found worldwide in the soil, in water, and most relevant to our discussion as part of the intestinal microbiota or microbiome. These bacteria, most of them, are very common to find in especially the lower intestine. However, there are some species belonging to the Enterobacteriaceae that are always associated with disease. These would be, as a real common example, something like Yersinia pestis. However, a lot of these organisms are actually going to be opportunistic. They're really only able to get in and cause a problem if the host is already compromised. So the majority of the species we'll talk about would technically be considered opportunistic. They are of moderate size and they do not form spores. Remember, spore formation is going to be associated only with gram-positive bacteria. Because they have that thick peptidoglycan wall, they're more able to easily convert it into the endospore covering. They are grouped in this family based on genetics, but also part of that is that they have a common antigen called the enterobacterial common antigen, meaning they'll elicit a very similar type of immune response. They are rapid growing organisms. In a laboratory setting, E. coli, which is one of the classic members of the Enterobacteriaceae, can replicate in 15 to 20 minutes. And they are considered to be facultative anaerobes, meaning they prefer oxygen, however, they do just fine without. And that's why they do really well in the lower intestine. They have pretty simple nutritional requirements, again, supporting the idea that they are relatively easy to culture in a laboratory setting. Finally, they are oxidase negative. That can help separate them from other gram-negative rods. If you are doing just basic biochemical testing, an oxidase test that gives you a positive result would likely be another type of gram-negative rod, perhaps Pseudomonas or Vibrio, whereas oxidase negative could lead you to the Enterobacteriaceae. The different genera belonging to this family can be separated by a number of different features. Most often, fermentation of lactose is going to be how clinicians will use selective and differential media to determine which member of the Enterobacteriaceae we're dealing with. They also can have resistance to bile salts, and they can vary in their ability to resist bile salts. And finally, the presence of a capsule. So in the Enterobacteriaceae, we call the capsule the K antigen usually. So the lipopolysaccharide, the O polysaccharide part is the O antigen. The flagellum is the H antigen. So strains will often be referred to using some combination of O, H, and K. The LPS is a major antigen found in the cell wall. We know that LPS can elicit an immune response specifically because of the lipid A or endotoxin component. Again, these different antigens, O, H, and K, elicit different immune responses. So the serological classification is often based on that combination of O, H, and or K. So, for example, E. coli, O157, H7. Most members of the Enterobacteriaceae are motile, and they have what we call peritricious flagella. So hopefully you remember your arrangements of flagella in organisms that have them. We can have monotricious, where they have a single flagellum, amphitricious, where they have it one on each end or a clump on each end, lophitricious, where they have a single clump on one end or another, and then peritricious, they're covered in flagella. So because they're covered in flagella, these are often highly motile organisms. Many of these organisms also have structures for adherence to the host. They're in the GI tract really commonly. That's an area where there's a lot of movement and potentially a lot of washing away. So it's really important that they're able to stick so that you can use their fimbriae to do that. These organisms are also very, very good at conjugation. So they can produce pili to help them exchange genetic material. Here's a list of some of the common virulence factors associated with this family. We'll talk about some, if not all of these, as we talk about the different species. So not all species are going to have all of these factors, but all of these factors are represented in at least some of the Enterobacteriaceae. What they're showing you here is what's called the type 3 secretion system. So type 3 secretion system. This is a really cool structure that basically resembles a syringe. It's like a molecular syringe. So here with the outer and inner membrane, we're trying to illustrate this is the bacterium. Here's a host cell membrane, and then here's the host cell cytosol. So this would be the extracellular space between the two cells. And this entire apparatus is the secretion apparatus. In this case, it's the type 3 secretion apparatus. And what happens is, as this is assembled in the inner outer membrane, it is then extended into the host cell membrane, and the bacterial cell can inject mostly proteins into the host cell. These can have a number of different effects on the host cell, eventually potentially leading to death of the host cell. So this is a really cool mechanism by which bacterial cells can go by and basically inject our eukaryotic cells. The most well-known and most prevalent member of the Enterobacteriaceae is E. coli, Escherichia coli, so E. coli. It is a highly effective pathogen. What you see here in this graph is a breakdown of Enterobacteriaceae infections. 45% are caused by E. coli. Very, very common. It is responsible for an estimated 80% of urinary tract infections, lots and lots of hospital-acquired infections, and what we most commonly think of it, gastroenteritis, or a GI upset. The reason that a lot of Enterobacteriaceae are associated with urinary tract infections is because the Enterobacteriaceae are often found in the GI tract. So they are secreted in the feces. In your patients with a vagina, what happens is with that particular type of anatomy, the urethra, which by the way is still different from the vagina, two separate openings, the urethra is closer to the anus and the urethra is fairly short. So generally what we're seeing with urinary tract infections in patients with female anatomy is that the patient has gotten a urinary tract infection from their own bacteria. With the penis, the opening of the urethra is at the end of the penis. It's much farther away from the anus, so it's harder. It requires much worse hygiene to get a patient's own fecal bacterium into the end of the urethra. Once we have catheterization, that can, of course, level the playing field, I guess, so to speak, and put both types of patients equally at risk for infection. And like I said, the majority of infections are endogenous, meaning the bacterium that is causing the infection came from the host's own microbiome. There are a number of different classifications of E. coli. This is a little bit out of date, but I'm going to go ahead and present it anyway because this is what is in the current edition of the textbook. There are two broad classifications of E. coli gastrointestinal infections, those that affect the small intestine, which we'll see on this slide, and those that infect the large intestine, which we'll see on the next. So we've got the strains of E. coli, the site of action, the disease, the pathogenesis, and the diagnosis. So the first is ETEC, or enterotoxigenic E. coli. Very, very common, 840 million cases per year, highly associated with consumption of contaminated food and water. This is often one of the major causes of traveler's diarrhea. So for patients who travel to countries that may not have the same type of sanitation that we have in the United States and end up with diarrhea, very, very common cause. The strain of E. coli, the ETEC strains of E. coli, produce two different kinds of toxins. Some are heat stable, meaning they'd be destroyed by proper cooking, but some are heat labile, meaning it doesn't matter. Either way, both of these toxins have a mechanism of action that lead to hypersecretion of fluids, which causes diarrhea. This is also the most common group of strains that cause infection in the US, associated with contaminated food. Enteropathogenic E. coli, or EPEC, is associated with infant diarrhea in developing countries, watery diarrhea and vomiting. It has toxins as well. These are mostly plasmid mediated, and what happens in this case is instead of hypersecretion of fluid like we see with ETEC, in EPEC, they have disruption of the structure of microvilli. So remember, in the intestines, first the intestines are super long, right, providing a lot of surface area. But then you have all of these microvilli, or little like pouches and projections that increase the surface area significantly. And the point of having all that increased surface area is for absorption of nutrients and water. When the structure of the microvilli is disrupted, you have a change in the amount of surface area for absorbing water, generally a decrease. The water has to go somewhere. It ends up being passed as diarrhea. Enteroaggregative E. coli is also associated with chronic diarrhea and growth delays in children. So this is something that I don't think we've talked about yet, but in many parts of the world, the leading cause of death in children under the age of 5 is diarrhea. Children are small and they're very prone to dehydration. So it is easy for a child who has developed diarrhea to go downhill quite quickly. The reason that we would see growth delays, again, what our enteroaggregative E. coli is doing shortens the microvilli. And when you shorten the microvilli, you decrease surface area, you have decreased fluid absorption, but you also have decreased nutrient absorption. So in all people, there's going to be a certain maximum height that a person could attain based on genetics in the absence of any sort of weird, tumors that lead to excess growth. So in an otherwise healthy individual, there is going to be a certain maximum height. And that is often dictated by the nutrient you've had access to as a child. Those of us who were fortunate enough to grow up having plenty of access to food, and you notice I didn't say who grew up in the United States, because there are a lot of people in the United States who don't grow up with access to food. Those of us who grew up with adequate access to food most often attain our maximum growth. Individuals who don't get access to nutrition, either because they don't have food to eat or because they can't absorb the nutrients from the food they eat, or maybe a combination of both, will experience significant growth delays. So while these two strains of E. coli are not terribly common in the United States, they do still exist, and they can lead to significant consequences when you have an infection as a child. Moving on to the large intestine, we have a Shiga toxin producing E. coli. So Shiga toxin producing E. coli, when I took this class, used to be called EHEC. EHEC stands for enterohemorrhagic E. coli. That might give you an idea of what one of the symptoms would be. Initial watery diarrhea followed by grossly bloody diarrhea. So the patient will be passing blood in their stool. This is because the Shiga toxins actually lead to not only shortening or changing the shape of the microvilli, but actually destruction of the microvilli. So the two Shiga toxins disrupt protein synthesis and basically kill the cells of the microvilli. So because you don't have the microvilli, you're not able to absorb water, so you have diarrhea. But because you're destroying some of the tissue in the intestines, blood can seep through, and so now you have bloody diarrhea. O157H7 is the most common strain of Shiga toxin producing E. coli that we see associated with disease, especially in the United States. Highly, highly associated with undercooked ground beef or any other foods that can be contaminated with feces, and this can include produce. An outbreak of O157H7 happened in 2009 in spinach that was harvested in Salinas, California, because of feral hogs that were defecating in the fields. More recently, you might recall that romaine lettuce has been pulled from the shelves with some regularity, it seems. Again, this romaine lettuce was grown in Yuma, Arizona. All of the lettuce fields were irrigated by a canal downstream of a cattle facility. So all of the canal water was contaminated with E. coli from the cattle feces, which contaminated the lettuce. And unfortunately, disease can be caused by ingestion of fewer than 100 bacteria. So you don't need very many bacteria in order to cause disease. The last type of E. coli is entero-invasive E. coli, EIEC. This is pretty rare. So again, you've got fever, cramping, watery diarrhea, may progress to dysentery with scant blood in the stool. So dysentery just means bloody stool. Grossly bloody diarrhea, you see the two terms used here. They kind of have similar meanings. Really what we're talking about is the idea that with grossly bloody diarrhea, you can actually see large quantities of blood in the diarrhea. With dysentery, it might be small quantities of blood, but it's still blood. And again, what we have here with entero-invasive, this is a little bit of a different mechanism than Shiga toxin producing. So the Shiga toxin producing E. coli, the S-tech, produce two cytolytic toxins that kill host cells. The entero-invasive actually invade and destroy the cells that way. So it's an invasive versus a toxigenic mechanism of cell destruction. As I previously mentioned, urinary tract infections are common manifestations of E. coli, generally originate in the patient's own colon and contaminate the urethra. In order for a strain of E. coli to be able to cause a urinary tract infection, it generally has to have some pretty good adhesins. There's a lot of flushing action in the urine, and sometimes hemolysins as well. So in this case, you could have a patient experience blood in the urine. In some infants, newborns, E. coli can cause neonatal meningitis. These strains have to have the K1 capsule. We don't know yet why this can cross the blood-brain barrier, but we know that it can, in neonates anyway. Otherwise, healthy individuals generally can combat this with their immune system. We can also see septic infections following E. coli. This generally comes from an untreated GI or urinary tract infection where the bacteria were able to destroy either the GI or urinary tract and get into the bloodstream.